speaker is Maria Konovalenko from Moscow. She will be talking about the need for a database that will unify the biomarkers of aging and other associated issues with that.
All right. Hello, everyone. And well, first of all, I'd like to start with thanking Aubrey and thanking all the folks at SENS Foundation for inviting me here, and also for putting up such a wonderful meeting. Please do carry on. We love SENS conferences. And of course, for the work that you're doing. So, in order to prolong lives, you need courage, you need a good plan, and you need a lot of money. Well, we've got a good plan. So the plan is to create an integrated information system on biomarkers of aging. And right now I'm going to tell you about what tasks the system is supposed to accomplish, why we need this, also what it may consist of, and what's the value of such a system may be. So, right now, well, aging is an extremely complicated problem. And right now, we have an enormous amount of data about aging. We know quite a lot about its causes and the mechanisms of aging. But there is a very well, serious problem that all of this data is very disconnected. And it prevents us from having a unified understanding of the mechanisms of aging. And also the process of creation of new therapies to fight aging and age-related diseases is also slowed down because of that. So the information system will allow us to systematize the fundamental, the basic knowledge about aging. Aside from scientific papers about aging research, there's also huge amount of clinical data about the age-related pathologies. We all know that, for example, metformin extends life, right? But it does so not in every mouse strain. And moreover, it sometimes extends maximal lifespan, and sometimes it just extends the median lifespan. Also, rapamycin was taken by quite a lot of patients who were recovering from kidney transplantations. And it would be a very good idea to collect this type of data about the impact of rapamycin on the health of these patients, because it may prove to be very useful when we will use rapamycin as a geroprotector, because we know it's a very, well, it's a potential geroprotector. Also, the system will allow us to model the processes of aging. And we know that each person is unique. Each person has a unique genome, a unique epigenome, metabolic peculiarities, and the system will allow us to identify the best combination of therapies that would cause maximal effect in this particular given person. So now let's take a look at what the database may consist of. So we see 3 steps in the process of creation of such well, complicated software product. And this task is a non-trivial task. So there are 3 steps. It's architecture creation, the software development, and database population. And so here are the necessary steps that need to be taken while creating the database framework. First of all, we need to identify where our information would come from. Obviously, scientific papers from PubMed, also electronic medical records, and already existing databases. I would like to draw your attention specifically to the importance of using electronic medical records. If we collect the information about a particular person and compare it to the scientific knowledge accumulated in the world by the collective, then we would be able to come up with really fruitful results for a given patient. And here's a list of existing databases, and I just highlighted the most interesting ones, like the AgeID, the GenAge, and the NetAge databases. So in order to get just the relevant information and incorporate and use it in our information system, we need some selection criteria, because we don't really want, papers about aging of metals or something like that, because it's not relevant to biology. So here's just a list of terms that may be used for this preliminary at this preliminary phase of information extraction, but maybe there are some other more specific terms which need to be defined. So the next really important stage is, all right, so we need to create the language, the dictionary that our system would use, and a set of terms that it will operate with. A colleague of mine, Savitskaya, well, she this is an example. She used the mouse genome informatic database, and she created a list of phenotypical terms. She searched for aging and premature aging in mice, and here's just a little part of that list that she retrieved. And if you take a look right here at the top, you can see abnormal nuclear morphology and abnormal nuclear laminar morphology. Well, that's the same. That's basically the same thing, and our system needs to know that this is the same thing, so we have to identify the synonymous terms. A really crucial moment. We need to identify the objects. So we decided to use qualitative and quantitative changes happening with aging, and use this information as the objects. So these can be classified. This can be divided into 5 groups. The first one would be changes happening over time, and they can be, for example, molecular changes like increase in lipofuscin in lysosomes. These type of changes can happen on physiological level. For example, a loss of undifferentiated stem cells in our peripheral blood. Another subgroup of aging changes is changes that we see when we compare our experimental group to control. Also, on molecular level, like for example, we can see that when we add thioflavin T to diet, there's a reduction in the amount of amyloid beta aggregates. And on the physiological level, we can see reduction in cancer incidence in rhesus monkeys. under caloric restriction, of course, and genetic changes. And here's an example of a genetic change that we would like our system to have. So obviously, changes need to be characterized. And here's a list of characteristics. Our system has to know in what tissue the change is happening, in which organism, and very importantly, the system has to know what the temporal and spatial values of the change are. And here's an example. There were two groups of people analyzed. The one was 64 years old, and the control group was 30 years old, and there was the authors of the paper looked at the peripheral blood mononuclear cells, and they found out that there's a decrease in this microRNA expression. And so this is just an example of how the change description may look like. All right, of course, changes are interrelated. And there are three types of links: causal, associative, and ontological. And here's a beautiful chart that a colleague of mine, Alexei Moskalev, created. He gave a talk yesterday about his work in Drosophilus in overexpressing stress resistance genes. So let's take a look here. We can see just an example of a causal link in black arrows. We can see that increase in these microRNAs leads to decrease of SIRT1 activity. And telomere shortening is associated with increase of these proteins expression. And this chart, it actually links events in our cells and in our tissues. Here, things that are increasing and things that are decreasing in our body. And here are pathologies and markers of pathology. So, okay, changes can be ontologous. We know that NRF2 gene has different names, and we know that the group of sirtuin genes, there's one of the genes in this group is SIRT1. So the last step would be to identify how we're going to retrieve the information. Well, obviously that's search, and the user can look for particular keywords or phrases. Also, he could choose everything that is in one category, like, for example, molecular cancer markers, or he can choose a term from the alphabetic ordered list. And of course, modeling, that would be a separate unit. All right. So we looked at the database creation. Now, just a couple of words about software development. The first step would be to create the domain-specific language, which can be used to describe. That could be, biologist-friendly, and could be used to describe events related to aging. And here's an example. There's Little B system. It was created in Systems Biology Department in Harvard Medical School, and it's written in Common Lisp, a computer programming language, and it does these things. Here's an example. It's there's EGF and its receptor, and a complex is formed, and so the system can actually model the temporal relationship between the concentrations of these molecules. So the main idea here is this is just an example of existing systems that can be used as a basis and modified in order to describe events related to aging. And database population, that's quite a daunting task since there's just enormous amount of data in the papers and in medical records. the extraction process has to be semi-automatic. So this means we just will use text semantic analysis tools, and there's a whole bunch of these. And well, inevitably afterwards, we'll need to go through all of that extracted data manually and verify it. So these were the 3 steps in database creation, and now let me just very quickly walk you through why we need this. So we will identify the biomarkers of aging. Right now, about 100 of biomarkers are used in clinic, and there are claims in the literature that 150,000 parameters can serve as potential biomarkers. We can model aging processes. This one is very important. Well, for example, just imagine, for example, we have a cure from aging. Let's say it's a combination of melatonin, rapamycin, and a magical compound X, right? And we want to test it in humans, because that's what we do. We conduct clinical trials, but we cannot really aid for like 80 years until the trial is finished, because if the experiment is successful, that's actually bad news for us, because we'll be dead by that time, or very old. And we need to know what kind of changes in which exact parameters will definitely tell us that a person will live for another 80 years, and these parameters would be the biomarkers. Once we've identified the panels of biomarkers, we'll be able to conduct longitudinal studies. And you all know the examples of such studies, like Framingham, Baltimore studies, and some of them are ongoing. For example, there's this Kronos lab, and these guys, they have panels of various markers, and they test them in a lot of patients. And it would be a great idea to collaborate with labs like that, because the amount of already collected data is tremendous and can be very useful. It is obvious that personalized science is one of the ways of life extension. So let me summarize. The integrated system of biomarkers of aging will allow us to systematize the existing data about aging. It will help us to systematize data about age-related diseases. We'll be able to model the processes of aging and interventions. We will also have criteria for assessment of the efficacy of interventions. This is the main idea of my talk. We can do this right now, well, after creating the information system, obviously. But anyway, we do have some very, very valuable information that can be applied to particular patients. And I'd like to thank my collaborators, Ekaterina Savitskaya, Alexey Moskalev, and Mikhail Batin, who is the head of Science for Life Extension Foundation, which I represent. And just very quick note, we're organizing the Genetics of Aging and Longevity conference in April in Moscow. And it's organized by the Gerontological Society of Russian Academy of Sciences, by the Science for Life Extension Foundation, by Moscow Institute of Physics and Technology, also International Association of Aging and Geriatrics, and Institute of Biology Common Science Center. So the topics include longevity genes in humans and model animals, genetics of regeneration, gerontology in silico, pharmacological interventions in aging, and quite a lot of other rather interesting topics. And the meeting is international, and anybody interested is more than welcome to participate. And I thank you all very much for attention. Thank you.
First question here.
very ambitious stuff. I'm wondering, in the examples you gave, it seems to me that there's an ambiguity between biomarker in the sense of an entity that changes in a steady way over time and gives you an idea about a person's biological age, and a factor that predicts a future rate of aging or negative health outcome. And of course, sometimes those things can be the same thing, but they aren't always, and sometimes they can diverge quite a bit. And a good example I'll use is IGF-1 levels, right? So normally IGF-1 levels fall with aging, but at least in animal models, low IGF-1 levels steadily throughout life predict a slow rate of aging. So which side of that divide do you want to break things down on?
Well, that is a good question. Whichever the processed scientific information would tell us to. I mean, the system.
Well, scientific information gives you both sides of that. That's why I'm asking.
Well, then. Well, in each particular situation, like there's not going to be a unified solution for a person, for a Drosophila, for a type of an organism. Every time the situation is really, really special, and when we have this ambiguity, we will have to take all of the existing information into account. And I think if we're looking at a particular person, we will be able to identify which side of those two ambiguous parties has more is closer to the truth.
It would seem to me what Pat had in the prior talk with his flagging of the ambiguous boxes, would be one of the solutions.
interpreted.
I mean, you can certainly flag it as
yeah.
currently unresolved. I mean, you're talking about the preponderance of evidence.
So I think this is a very laudable effort, and I would just like to point out that the C. elegans bioinformatics community had set a very similar goal within just this one simple, less than 1,000 cell organism, and they've yet to realize it, to come even close. But you might learn something from the model that they created and the difficulties that they've encountered.
We'd love that. Yeah.
You must obviously do the same thing for each organism, and in a relational database, you can link the different organisms to see where there are parallels.
Yeah,
it would be very nice.
Sure.
Greg?
I commend you on your link to the primary literature in your database. I think that's essential because that's what we all need to evaluate in terms of the quality of the data. It seems also, I'd like you to comment on whether you're going to need a separate database for every organism with aging, and then within mammals with a separate database for organs, because, for example, gluconeogenesis, which may go up in the liver, goes down in muscle and brain with age.
Right. Well, when I was talking about the objects, the change. So we observe a change, and it's got the classificators, right? And there's the organism and the type of tissue. So we are. Some changes can be duplicated, but they would, since they have different classification fields, they will be all in like one big jar of information, right? But the system will be able to retrieve everything relevant to C. elegans or a human, and it would look at the classification fields, and therefore it would not, mix the right and wrong information in each particular situation.
Okay, any other questions? There's one over there. It's a final question.
Well, just a clarification question. Is your objective here to actually model and predict the behavior of the human body, for instance, during aging?
Yes.
Or is it as a reference manual? You actually intend to make predictions?
Yes, we would like to predict. We'd like to identify what the magical combination of therapies would be ideal for this particular person. Yes, we are. That's the goal. Probably it's like a long-term goal because we would like to have some differential equations that would describe the processes, but as Pat has mentioned before, we cannot really do this right now.
Then essentially the same question as asked the previous speaker. The accuracy of any prediction made would be heavily dependent on the complexity of the underlying system, and if you actually intend to model the pathways, the task would be enormous. So what level of accuracy do you expect you can achieve without like a complete matrix explosion underneath?
Well,
maybe
the best
that's
possible. Would that
possible to
count as
answer.
an answer?
Okay.
Well, we would basically, one patient, one person would have an ideal situation is to have the information system and a couple of, biogerontologists attached to the system that would, at least at the first stages, verify the quality of the predictions. But eventually, the goal is to, make the perfect predictions, whatever they may be.
Thank you, Maria.
Thank you.